I'd like to welcome everyone to our second webinar of our seminar series that we're proud to have started. Today's webinar revolves around small molecule delivery systems via microfluidics. I'd like to first start off before Ian starts off, and we'll end with you, Ian. I'd like to start off by introducing the West regional team with our Southwest field application scientist, Viet Nguyen. That's Viet. Our regional sales manager for the Southwest, Kim Killian. I'm Angel Galarza. I'm the regional director for the West. Thank you again, and we'll head over to Ian Villamagna to start off with the actual presentation. Ian?
Great, thanks, Angel. Appreciate that. And so, yeah, thank you for joining us, everybody, for our second Western Regional webinar here from Precision Nanosystems. And today we're going to be looking at small molecule delivery systems that are prepared with microfluidics. So the goal for today is really to go into a background of small molecule delivery systems, some key examples of them, the different ways that they look, and then some of the challenges that are associated with small molecule delivery systems that are kind of, in a sense, holding that field back, and how we can maybe work to get around some of those issues that we see. So our vision as Precision Nanosystems is to accelerate the creation of transformative medicines that significantly impact human well-being. And I think what really embodies that vision are the things that we're doing today. So these webinars that we do are a means to communicate with our customers and to share the expertise that Precision Nanosystems has across the board throughout all of our organization, and share it with our customers to help drive the formation and the creation of these transformative medicines that our customers are creating. Small molecule drugs, I think we can get into a sense now where we're forgetting about small molecule drugs very easily. And part of that is because we're hearing a lot lately about nucleic acid medicines or biologic drugs. But it's important to remember that even today, small molecule drugs continue to make up the majority of the current pharmaceuticals on the market. And so if you look at something like in 2017, the majority of new molecular entities that were approved by the FDA were still small molecule drugs. And so while we've seen this massive trend towards biologics and towards nanomedicines using things like nucleic acids, like RNA or DNA, small molecules still make up a large portion of what we're dealing with in terms of medicine development. And really, part of the reason for that is that these small molecules can be used to treat a myriad of disorders, things like cancer, things like infections, inflammatory disorders. And so they're very useful to us still as a tool in medicine. Part of the reason also that I think you'll never fully see small molecules leave the medical space is that the development of small molecules is typically much easier than the development of something like a large biologic molecule. And so if you think about something like aspirin, where you're dealing with a molecule that is only comprised of 21 atoms, and compare that to something like a large biologic where you're dealing with over 25,000 atoms, what you have is a drug that was much easier to manufacture and develop than something like a large molecule drug or a large biologic as well. And so I think there's a lot of history and there's a lot of data with small molecule drugs that we can lean on. And it allows us to develop these small molecule drugs continually in a way that's easier still than developing these biologic drugs. And so that really rolls right into the benefits of these small molecule drugs. They are relatively simple to manufacture. And part of that is because there are so many protocols and procedures that are already in place to develop these drugs that we can utilize those again and again to develop different drugs. Another thing to keep in mind with small molecules is something that we see a lot of, and that is generics and biosimilars. And so what we're able to do now are take small molecule drugs that have been utilized before and essentially repurpose them as a biosimilar with very small changes in the actual chemistry of that small molecule and utilize them again. And this is very common, and it also works to increase that amount of small molecule drugs that are on the market. From a physiological standpoint, a lot of the pathways within the human body, the physiological pathways, are actually driven by small molecules, biologically derived small molecules. And so from a logical standpoint, it does make sense that we're still going to utilize these small molecules that we can develop in a laboratory to try and mimic what our own bodies can do on their own. There are still challenges that are associated with small molecule and small molecule drugs, and these have been present really since the start of medicine and since we started using these drugs as treatments. And I'd say probably the first one that's most common and the easiest one to talk about is systemic toxicity. So when we're using these small molecule drugs, how do we use them in such a way that they're going to have a benefit for what we are specifically trying to get them to do, but not be systemically toxic where they're causing issues in other parts of the body? Some other challenges that occur with these small molecule drugs would be the bioavailability or the half-lives of these molecules. These molecules are very small by nature, and so putting them into a body, they will get essentially rapidly filtered out of the body. And so how do we get it to a point where we can deliver these drugs with a high bioavailability so they're able to do their job effectively? Furthermore, the efficacy can really rely heavily on the therapeutic target. And so, it goes back to those physiological pathways, but there are some places that these small molecules can work very well, and there's some places that just do not work as well because they're not as able to do their job. If we begin to look at why a carrier for these small molecule drugs are so crucial, it's helpful to kind of look at an example. And I think a lot of times when we talk about small molecules in the industry, what you're going to go back to is the example of Doxorubicin, because this is a really good example for a lot of reasons, and it's still very much used as a benchmark for our development. So Doxorubicin is an anti-cancer agent that's been around for quite a while now. It was first approved in 1974, and it works through the inhibition of DNA and RNA synthesis, through inhibition of topoisomerase. But what was really quickly elucidated after we started using Doxorubicin is that it was also causing essentially targeted apoptosis in cardiac myocytes. And so it was really leading to this cardiotoxicity that was really detrimental to the people that were taking it. And it was a difficult, it's a difficult thing to deal with still, because Doxorubicin as a whole works very well for what it's supposed to be used for. So for anti-cancer agents, Doxorubicin is still extremely effective, but the toxicity is something that's had to be balanced since it's been approved. And how do we use Doxorubicin in such a way that we can balance that toxicity, or even still use it knowing that the toxicity is going to come along with its use? The kind of answer that came out of Doxorubicin was Doxil. And so Doxil was actually the first quote unquote nanomedicine that was approved by the FDA. So this is a liposomal formulation in which they took Doxorubicin and packaged it within a liposomal construct. And so you're looking at an image on the right here of that construct. And so what you see in the circular, in the circles, is actually the liposomes. And then within the liposomes, those stripes essentially down the middle, is the crystallized Doxorubicin inside of that liposome. And Doxil did successfully encapsulate this drug molecule within the carrier. And it allowed this to be delivered directly to tumors, which limited the systemic toxicity. Doxil as a whole was really a wild success in a lot of ways. It did very effectively limit that systemic toxicity and gave us the ability to use something like Doxorubicin down the line, and without that systemic toxicity causing widespread issues in the body. And so still today, Doxorubicin and Doxil serve as a really important benchmark in the development phase for medicines, and especially for nanomedicines that are using these sorts of carriers. So, Doxil in general was revolutionary in a lot of different ways. And if you look at a bunch of studies that are looking at things like liposomes and the encapsulation of toxic molecules into carriers, they will often compare themselves back to Doxil as that benchmark and see how they're working against that Doxil benchmark. To say that it was approved and used and never ran into issues, though, would be disingenuous, because Doxil was hampered with quite a few issues that remained after its approval. And one of the biggest ones that actually came up with the production of Doxil was that they just couldn't essentially manufacture enough of it. There were shortages for a lot of years of Doxil. And there's a couple of different reasons for that occurred. And those are in these three boxes down at the bottom. Really, the first reason came down to the liposomal production. And so essentially, when you're making these liposomes, what they were finding is that they were getting these giant vesicles rather than just singular liposomes like you saw in the image in the last slide. And what you have to do at that stage is actually take these constructs that you've made and then break them down into smaller constructs, into singular liposomes that can be used for the drug delivery. And that can be challenging to do. It can be difficult to do that, and it can be difficult to get a homogeneous population from doing that. Another challenge that's associated with Doxil is that after these liposomes are formed, so after you get past these giant vesicles and you have your population, you still have to remotely load this drug into the liposome. So you actually have to take the drug from outside these liposomes and load it within the liposomes through chemical reactions. It's doable. It's been shown many times, but it still has challenges associated to it. And you can imagine that the challenges that you see, especially at something like a laboratory scale, are typically magnified quite a bit when you're doing this at a large scale for a manufacturing production. And so, continuous scale up really wasn't seamless, and they saw that right away. When you are making liposomes and doxorubicin loaded liposomes on such a large scale, you're usually using these large scale machineries. And what they quickly found is that it was very difficult to continually manufacture and produce these liposomes with doxorubicin in them in such a way that they had a quality product. And so in roughly 2012-ish, I believe, Johnson & Johnson, who had essentially brought Doxil through the FDA and through clinical trials, voluntarily stopped selling it because of manufacturing issues that they were running into. And so when you think about manufacturing these drug delivery systems, it really becomes important to think about manufacturing them with an effective method. And so this is really where something like microfluidics can come into play and is very important. The difference in microfluidics compared to something like a traditional approach for liposomal formulation is really that it's completely the opposite. So whereas something like a traditional formulation of liposomes would use extrusion, for instance, something that's a high energy top-down technique where you're starting with these large constructs and breaking them into smaller pieces, microfluidics employs an approach that is bottom-up. So you start with your pieces separate in different fluid streams, and by mixing those fluid streams, you're able to form controlled liposomes or particles that are the same size, they're a homogeneous population overall. Microfluidics as a whole has changed quite a bit over even just the past 10 years. And so you can look at things like here on the right, which is next-gen microfluidics, which is kind of the next logical step from microfluidics. It takes that mixing path and retains the physics of mixing through those mixing paths that you would see in more traditional mixers, but allows you to scale these constructs much larger. And so with something like next-gen microfluidics, you're allowed to take your constructs and scale the production of them to the point where you can make millions of doses very quickly, and still with that same assurance that you're going to have a homogeneous population overall. And so we're going to kind of shift gears now a little bit. That's a little bit of background. And now what I'd like to do is actually kind of focus on two of the main delivery systems for small molecules. And so that's going to be liposomal delivery systems like Doxil, and then polymeric delivery systems as well, which are very important and gaining a lot of traction, I think, in the field. And so we're going to kind of shift gears into that, starting with liposomal delivery systems. So liposomal carriers are essentially made up of this vesicular structure where they would have a bilayer in them. And so what you're looking at here in this figure on the left is actually a singular liposome that would have been formed after it had gone through any sort of breaking down of those multilamellar vesicle structures if you were using extrusion. Using microfluidics, you're able to make these singular liposomes on their own the first time from passing your starting materials through that microfluidic channel. The components of liposomal carriers can vary based off the formulation and the encapsulation of what you're trying to encapsulate, but typically the components stay somewhat close to what's listed here on the screen. And so you would typically have a pegylated lipid around the outside to keep it stable and keep it from aggregating into other liposomes. You'd have helper lipids to actually fill out the bulk of your liposome and have structural effects on that liposome. Cholesterol, again, is another structurally based liposome that helps to increase rigidity of that membrane. You have either the ability to use hydrophilic or hydrophobic drugs, including amphiphilic drugs. So liposomes are really important in the sense that they have a wide variety of what you can encapsulate into them because you have that bilayer and because you have that hollow chamber essentially in the middle of your construct. You're able to load different drugs into it. And then the final component that you see in teal, which is probably the most commonly spoken of component, would typically be something like a cationic lipid, which actually forms the bilayer and then folds into these liposomal structures. And so looking at some key examples of liposomal production, what you can really start to see is that liposomes can be used really effectively to limit things like systemic toxicity. And so this is true not only with doxorubicin, but with other constructs as well. And this paper here, a 2019 paper by Hamano et al., was really interesting in the sense that it showed the loading of curcumin within a liposome. And it showed, firstly, a more prolonged release of the curcumin and circulation in the body, which is extremely important in the sense that the longer you can circulate these drugs while still limiting the systemic toxicity, the more likely they are to be able to work. But what I think this really showed really effectively as well was this figure on the left here, which is looking at essentially a measure of kidney damage after administration into an animal model. And what was interesting here is that they weren't actually looking at the effect of the loaded cargo and the toxicity of the loaded cargo here. They were actually looking at the effect of toxicity on another drug, cisplatin. And so cisplatin is used as an anti-cancer agent as well. It's extremely toxic. And what they were able to find is that by actually using cisplatin in addition to a liposome that had curcumin loaded into it, through using that combination, they were able to reduce the kidney damage they were seeing overall, compared to when they just administered cisplatin alone. And so they have a couple of instances within that paper where they try to elucidate the mechanism. But it's an interesting paper in the sense that you can see the effects of having a drug-loaded liposome that plays an effect on the overall toxicity of a drug. Another really interesting example of a liposomal formulation for small molecules comes out of Yvonne Perry's lab at the University of Strathclyde. And so Yvonne Perry's lab does a lot of work with liposomal formulations and small molecules, but really drug delivery systems as a whole. And so in this paper by Yoshi et al. In 2016, they looked at actually including two different drugs into these liposomal constructs. So a lipid-soluble drug within that bilayer, and then a water-soluble small molecule within that hollow chamber, essentially, in the middle of your liposomal formulation. And what's different here than what you would see with something like Doxil is that it was not done through remote loading, but it was actually done in situ. So they were able to put the components right through that microfluidic chamber and through the cartridge to actually form their liposomal constructs with the drug included into these liposomes at the end. And what you can see first and foremost is really interesting is that through doing it in this method, through not using that remote loading, you do still form these liposomal constructs and you do form them readily. And the size, when you actually include this drug, tends to go down from a non-drug loaded liposome. So you can see on the bottom here, the typical size for a non-drug loaded liposome was at about 80 nanometers. And through adding drug, either one or both, you're down into a range that is closer to 60 nanometers. If you only have one drug, it's typically a little bit smaller. The other thing that was interesting is really, if you look on the right, the drug loading amount, compared to when you're just loading one drug alone, the amount that you can load of the two different drugs does not statistically change if you're loading two different drugs. And so essentially what that showed is that the capabilities to load one drug into that bilayer are not affected by the capabilities that you have to load the drug into the center of your liposome either. And so you're able to actually load two different types of drugs and have a double action sort of liposome that can be used overall. I would recommend to anyone that's interested, double checking with Yvonne Perry's lab. They put out publications all the time, and they essentially continue on this pathway and have a lot of different interesting small molecule delivery liposomal formulations that are interesting approaches and interesting follow-ups to this sort of work. So changing gears now, we're going to look kind of at polymeric small molecule delivery systems. And so this is something that is close to my heart. This is kind of what I did my PhD on. And so polymeric system is different from the liposomal system in the sense that typically what you'll see with these polymeric systems are these solid core particles. And so they're made up of essentially less components. They're typically simpler overall, where you'd have something like a polymer, which you see here with white strands, a surfactant, which you see in blue, which helps to stabilize the particle. And then what you're seeing here with these little red dots are essentially the drug that is loaded within these structures. And the drug in these cases is typically loaded throughout that particle, not stuck in one spot or not just on the surface or in the core. It's typically loaded homogeneously throughout. And there's a lot of really interesting publications as well using these polymeric systems that are great instances to look back at and try to understand more about how these delivery systems can affect things like toxicity. So this is a really interesting paper that came out in 2019 where they looked at a potential drug candidate where they had a high metabolic stability. So it looked like it was going to be a very useful compound to be using. But it did also have a very high potential toxicity due to the way that it programmed cell death. And what they were able to see is that through encapsulation in a PEG PLGA nanoparticle, they were able to significantly decrease the toxicity, which you can see on the right here in this graph. So in both green and red, what they were able to see is after treatment in an animal model, 13 days post-treatment, they looked at the weight change as a measure of toxicity overall for that animal. And when you deliver this molecule without a carrier, you do see a significant drop in the weight of these animals. But Angel?
Sorry, we have two questions on the previous, I think it's on the previous slide. Would you mind reading those questions, please?
Sure, absolutely. They're somewhat related. That they refer to the components of the liposomal formulation, and the question is, how do lipids used in the liposomal formulation, Ian, would affect the drug loading, and can we load two drugs, hydrophobic and hydrophilic, together by an in situ method?
Yeah, so I guess as a whole, the change in lipids is going to affect the formulation overall, depending on the drug that is typically being used. And so I think, from a formulation standpoint, where we would typically start in developing these formulations, is to start by looking at the cargo that we're encapsulating and trying to understand the type of lipids that we need to actually build around that cargo and effectively encapsulate that cargo. And so that kind of leads in, like you said, Viet, to the second question, where, can we load something like a hydrophobic and a hydrophilic molecule? Off the top of my head, I can't think of a specific example where I've seen that publication. I'm sure it has been tried. I'd have to look back and look for a specific publication where someone's done that, or a specific instance where someone's done that. But I think the potential is there. And I think, the other thing that is important to keep in mind is that, I know we talk a lot about this sort of remote loading versus in situ loading. you're not necessarily in an instance with these liposomal formulations where you can only do in situ loading and it's 100% better every time. There are instances still where remote loading can be very effective. And so, potentially it could be an instance where you're looking at something like in situ loading and remote loading of your liposomes, which could allow you to do something like that. But that's kind of a formulation question. It would be dependent really on what you're trying to encapsulate and the lipids you're trying to encapsulate as well.
Thanks, Ian.
Yeah. Thank you, Angel. Thanks, Viet. And so for this this carrier that we're looking at here on this slide, like I was saying, the incorporation of that small molecule into the nanoparticle, which you see in blue, didn't significantly cause a change in the weight of these animals after administration, leading us to believe that it was less toxic than just delivering that small molecule alone. This publication came out in 2018, and so this one was very interesting. This came from the Ferrara lab at Stanford University, and what they were actually doing was comparing a small molecule that had actually already been altered, trying to make it more bioavailable, and compare that to nanoparticle availability and bioavailability after administration. And so what they did was actually squalanated their small molecule. And so what that does essentially is it takes a large chemical compound that has natural origins and adds it to that molecule. And the belief is that the increased size should increase the bioavailability and the circulation time of that molecule. What they did is they actually went one step further and they compared that squalanated molecule to a nanoparticle that was made with that squalanated drug in it. And so what they were able to see is that they were able to actually create nanoparticles with that drug molecule that had that squalene group on it. And through biodistribution studies, which you see in the middle here, they did see the nanoparticles in red had a longer circulation time in blood overall and in other organs as well, showing that it had a better biodistribution overall. But I think really what it came down to, what was a really interesting figure in my opinion, was the whole body radioactivity after 4 hours. So here they took a radioactive compound and put that within their nanoparticles or onto the squalanated drug. And after 4 hours, they saw a statistical difference in the amount that was still circulating in the blood, showing that, you can do things like drug modification, but there still is a benefit potentially to using a carrier system like a nanoparticle to even go beyond what you can do by just altering that drug molecule and having further effects on things like the bioavailability.
Ian?
Yes.
Just a bit of a follow-up question. Having seen some of these liposomal and polymeric systems, we had a question regarding if you're looking to encapsulate the hydrophobic molecule, how would you choose whether to go with, say, a liposome or a polymeric particle, or even solid lipid nanoparticle?
Mm-hmm. Yeah, that's a really good question. And I think part of that typically comes back to the experience that the individual user would have in their laboratory. And so why I say that is, typically what we find is that customers who come from polymeric backgrounds are typically very polymeric, and customers that come from more lipid-based backgrounds will typically be more likely to use something like a liposome or a lipid nanoparticle. The nice thing is really, and I think it goes back to the nanoassembler platform for microfluidics from Precision Nanosystems, is that you can make liposomes on the nanoassembler platform. You can make polymeric nanoparticles. You can make, like you said, via solid lipid nanoparticles as well. And so you really do have a wide range of abilities to do that. And it really kind of depends, I think, on what your end target is, and what kind of sizes you're looking for and what kind of effect you're looking to have. And so there's benefits to things like polymers that you necessarily can't replicate with liposomes, especially in things of like controlled release or prolonged release that you don't necessarily get with liposomes. But then there's also benefits to things like cellular uptake with solid lipid nanoparticles or liposomes that can be hard to mimic with polymeric systems. And so I think looking at the actual end goal will kind of dictate where is most logical to start. But you are able to kind of look at a wide variety of different places to start. And you have the capabilities and abilities to do that across the board with all sorts of different types of cargoes.
Thanks, Ian.
Yep. And so our last publication about polymeric delivery systems for small molecules is really kind of one that I go back to a lot. And so, you know, for anyone in this webinar that has seen me speak before or that I've visited and has listened to my talks, I go back to this figure quite a bit. And the reason is because it really shows how important it is to be able to create particles of a certain size. So here, this is a paper from 2015 where they actually looked at photophytotoxin, which is a drug that is known to circumvent cancer drug resistance. So it is highly effective and it has a lot of potential use, but it is extremely toxic. And so it does limit its ability to be used. And so what they looked at here in this paper was actually using a PEG to conjugate that drug and then forming polymers from a cellulose backbone. So a very naturally derived polymer system to make these particles. And what they really wanted to get an idea of here is how deviations in the size of these particles would affect things like the pharmacokinetics and the biodistribution. And so this figure on the right is really, I think, important in showing the absolute importance of having control over that particle production in terms of size. And so you can see that after both 48 hours and 96 hours, particles that were sized in the 20 nanometer range did selectively end up in tumor tissues where you would want them to be for that drug to work. But when you compare that with something like 120 nanometer particles, you saw a lot of different distribution of these particles within things like the liver or the spleen. And so getting to different parts in the body where you don't necessarily want them, you want these concentrated in the tumor. And so what this really goes to show is that having that fine control over these particles and the development of the particles can be extremely beneficial in actually targeting the tissue that you want to target rather than just delivering this for systemic toxicity. And so that kind of is a really efficient segue actually into just the final portion of this webinar. And that's actually utilizing these microfluidics to prepare these small molecule delivery systems. And so how do we prepare these systems in such a way that we can control the size and the final constructs?
Ian,
Yep.
Before you go ahead, could you move one slide back? We have a question about the size dependence on distribution from Sarah. And the question is, do you think the size dependence seen in this publication could have other causes, or do you think it would extend to liposomes as well?
So is it specific to the fact that they used this polymeric system rather than liposomes? I think, you know, from what we've seen, we do know typically that smaller constructs will aggregate in tumor tissues more readily. I think this has been seen, and I can think of a couple of publications from recently that I spoke about where, you know, we saw this with particles like this that were made from polymeric systems. I'm pretty sure I'm thinking of one specifically that is a liposomal formulation as well. You know, I think it kind of can get back to the EPR effect, where you're essentially saying that these particles of a certain size will be more likely to end up in tumor tissues. The EPR effect is really widely, I don't know if disputed is the right word, but it's not widely accepted. And so I'm hesitant to start going into EPR effect there. But I do think that there is a definite relationship in the size of the particles and where they actually migrate to once they're used in the body. And that is true regardless of whether it's liposomal or something like polymeric.
Thanks, Ian. And a quick follow-up to a previous question, actually. Would you think a polymeric system is more is better for a sustained release of small molecules in vivo? And how long can we maintain these drug releases in that case?
I don't necessarily know that you can make a blanket statement and say that polymers will always be better for a prolonged release in vivo. What I do know, just from my own history, is that polymers give you an ability to really tailor how they degrade rather than relying on something like a lipid system where they degrade fairly readily. And so, depending on the polymer you're using, and there are a lot of different polymers that can be used and that can be used to make particles, you can manipulate that polymeric system that you're using in order to really prolong that release. And I think that, in my opinion, is one of the hallmarks of a polymeric system that really changes as compared to something like a liposomal system where you're using that lipid-based component. There are still methods to essentially use those lipids and potentially to prolong the release from lipids. But I think just from a matter of what's been seen before and places to build off of, polymers and the ability to manipulate those polymers gives you a really wide range of abilities to do that.
One more formulation type of question from, sorry, I think the name is a bit too long. I won't attempt to say it. In your opinion, a peptide-based drug will be best in a liposomal type system or a polymeric system?
I don't know that I would make a claim as to what would be best. I'd have to actually look at the specific formulations that were made. I think what I can say is that I have seen peptide-based systems more readily with liposomal formulations. That's what I've come across more, especially recently when I've been looking at things like this. But to say best versus polymeric, I don't think you can necessarily make those sorts of claims. I think the biggest limitation with a polymeric system and something like a peptide traditionally is the solvents that you have to use to actually form those particles. And so you have to make sure that you're using a solvent that is not too harsh for that peptide, that could cause changes in that peptide or something to that effect. And so I think that's one of the things to think about with a polymeric system and peptide delivery systems.
Thanks, Ian.
And so, this last kind of chunk here, the last couple slides, is really about utilizing microfluidics to prepare these delivery systems. And it really ties back into the issues that they saw with Doxil production back when it was first being manufactured, and how they had a really difficult time scaling up. And so, we talked about this at the beginning with microfluidics as a whole, and essentially how microfluidics gives you that exquisite control over your particles that you're making. And so microfluidics traditionally would utilize two fluid paths flowing into a mixer to mix your two fluids and actually form your particles through self-assembly in that mixing path. And traditionally, you'll see things like this staggered herringbone mixer, which are really effective, and they've been used quite readily for many years. The issue that remains with something like a standard herringbone is that the mixing is actually controlled by these chevrons that you see in the middle of this mixer. And each one of these chevrons is a 3D structure. And so when you go to scale that into a larger system, it can be difficult to do. And we talked about this a little bit before, but next-gen microfluidics basically maintains that same mixing of the fluids, but without those 3D structures, allowing you to scale the geometry of that mixer in such a way that you can scale the amount that's going through it and the amount that you're able to produce. To get an idea of how the scalability works, we have a couple studies that we did recently looking at liposomal formulations where we encapsulated verteporfin, so a small molecule drug, into a liposome. And what we were doing here was actually comparing the production of these systems on a benchtop scale, so doing things at a slow flow rate, versus a blaze system, so a larger pre-GMP scale where we were running these constructs at a much quicker flow rate. And what we saw across the board is that the particle size and the polydispersity remain constant when you're using this next-gen mixer, whether you do this on a benchtop scale at 2 milliliters or a blaze system, a larger scale, at 20 milliliters. And so we saw the exact same thing as well through encapsulation efficiency, which is really important because the size and PDI is important for liposomes, but the encapsulation, the amount of drug that's actually being included into those constructs remains important as well. And so being able to scale that in such a way that we can not only include or see no change in the size or PDI, but also the amount of drug that's within those constructs is very important. And we're able to see that using this next-gen microfluidics. And so this really brings us back to the idea that you can use these next-gen microfluidics to really scale these small molecule delivery systems up very rapidly and very reproducibly. And so this graph that you're looking at here really shows across the range of how you're able to do that. And so what you can do essentially is if you develop at something like 12 milliliters per minute, so something that would be a very typical laboratory scale, what we've shown is that you can translate that using next-gen microfluidics all the way to a GMP system where you're running at something like even 90 mils a minute or 200 mils per minute and create particles that are the same across the board because of that architecture that we're able to scale for the microfluidics. And so what that really means, I think, as a whole, is that using an approach like this will give us the ability to scale the production of these small molecule delivery systems and not run into those issues that we've seen in the past with something like Doxil production, but reproducibly scale this up so we can make millions of doses that are needed very quickly, but also very effectively and maintaining that same homogeneous population that we see in a laboratory scale.
So Ian, somewhat related on the scales, there's a question. That's basically asking that based on your expertise in drug delivery, and knowing how I guess drugs for coronavirus are delivered, what are your views on the potential of small molecules to combat the virus, or as a treatment, or as a?
Yeah, I mean, I think small molecules to combat the virus, in my opinion, would be mostly in terms of a treatment. So, I think it can be really easy to get caught up in COVID and to forget that a vaccine is preventative and a treatment with a small molecule is just that. It's reactive. So it's how do we stop this once it's actually started rather than how do we stop this before it starts. But I do think actually in both instances where microfluidics comes into play and where this sort of data that we just looked at comes into play is that ability to scale up very quickly. And so this is something that we're seeing. Like if you think about Moderna's vaccine that they're pushing through clinical trials, when you utilize something like an mRNA vaccine that has a lipid nanoparticle carrier, or really anything that has a delivery system that has to be made, when you can utilize microfluidics to do it, and you can utilize a system that is able to scale up that rapidly, it de-risks that step into that manufacturing process. So that the stuff you did in the lab to actually develop that drug, be it a small molecule drug with a carrier, be it an mRNA vaccine with a carrier, that readily translates into the GMP scale at that speed at which you need. And what it really brings us the ability to do is deliver a large amount of doses very quickly, which we know, I think, as a whole right now, is going to become even more important down the road to combat things like COVID-19, where you need a large amount of doses to actually stop the problem before it can get worse.
And somewhat related question on manufacturing would be, what is the effect of the manufacturing method then on the stability of these constructs you make, be it, I guess I'll make it more general, liposomes or other types of nanoparticles?
Mm-hmm. Yeah, I mean, I think that, I'm maybe not the best person to speak manufacturing here. This probably goes, admittedly a step beyond where my expertise kind of is. But, I think from a general standpoint, what we know is that, the manufacturing methods can have an effect on the overall stability of your constructs. So, if you think about something like extrusion to make liposomal carriers and formulations, what we know is that the stability coming off of a liposomal formulation from extrusion can essentially be less stable than something coming out of a microfluidic based system, partially due to the fact that you don't have as homogeneous of a population. The other kind of avenue to remember is that if you're creating these populations with a system that is not giving you a homogeneous population from the beginning, what that means is that you have to do more downstream processing to actually get the formulation to where you need it to be before it's a usable drug product. And so that can mean essentially more things like tangential flow filtration, using longer flow filtrations, different sorts of extrusions to actually break those particles down and make them into what you need them to make. And each step you add to that process adds a different layer of complexity, which can affect the overall formulation at the end. And so I think what it comes down to, in my opinion, is that if you can use a system like microfluidics, where you really go and have your particles come off and your constructs, your delivery systems come off as ready as they can be from that production system, then it also helps to make your downstream processes much easier. And it just kind of accelerates the entire process throughout the way.
Thanks, Ian.
And so just in conclusion, really, small molecule delivery systems are widely utilized in the pharmaceutical industry. And, I think the notion that small molecule medicines are going to disappear with biologics or be completely replaced with RNA medicines is not the case. I don't see that. I actually more envision a scenario in which we leverage what we know about delivery systems to better use these small molecules and use them in conjunction with things like nucleic acid medicines or biologics to really more effectively treat diseases from the start. And so I think, if you're thinking about encapsulating with a carrier, what it can really come down to first and foremost is reducing the toxicity. I think that there are a lot of publications, there's a lot of instances that we can point to where people have done studies about the reduction of toxicity through using these carriers. But they also can affect things like the biodistribution and the availability, and even things like targeting to specific tissues, which we didn't get into today, but there is that capability as well, as a whole different avenue that can be explored through the production of these delivery systems. And I think really the final kind of takeaway is that microfluidic parameters can really be utilized to try and essentially de-risk that situation that you saw with something like Doxil, where you had that production issue from the start and it led to a significant delay in the production of the drug, which was needed very rapidly. And so using this microfluidic pathway, you're able to actually create a more available and ready drug product than you would through other methods.
Ian, a question to the point you really just made about the targeting. As a follow-up to the previous peptide encapsulation question, whether a liposomal system can influence capacity of the drug to pass the blood-brain barrier?
Yeah, I think it has the potential. I think what you can do with liposomal formulations is typically add things to them that specifically allow for targeting. And so this can be done, for instance, just off the top of my head, you can use lipids that have antibodies conjugated to them. You can actually conjugate typically other molecules to lipids before you create the liposomal constructs. And what that allows then is those liposomes to take on essentially a different sort of delivery method, where they're able to either attach to specific sites in the body that you've targeted, or potentially do things like pass that blood-brain barrier. We didn't really get into that today. I think there are definitely some examples of that. And so if you're interested in that and any specific examples that we've seen with the nanoassembler platform, you can feel free to reach out to us and we can make sure that we point you in the right direction for publications that we know of that have been used in the past as well.
Thanks, Ian.
And so just with that, we'd like to thank you really for attending our Western Regional webinar this morning. Any questions that you have, Angel and myself cover kind of the Northwest area, Viet and Kim cover the Southwest. If you're unsure of what region you actually fall into, feel free to reach out to us and we will point you in the right direction. Alternatively, if you're joining us from outside of the Western region today, we do appreciate you joining us. And if you're interested in learning more, you can feel free to reach out to any of us again, and we will point you to your regional team in the area as well. So with that, thank you for your attention, and any follow-up questions I'm happy to take now. If not, I'll turn it back over to Angel.
Thank you, and really appreciate that. I'd like to share some upcoming events. Many of you already know of our journal club. I've seen some familiar quote unquote faces joining us during our webinar this month. Please note that these are monthly. If you're interested in any specific topic, please feel free to forward along. We're more than happy to address specific topics or add it to a general topic that we might be speaking to and on. But that being said, we also have something called our journal club that we have on the second Friday of every month. We've already done a couple of them. We're going to be doing our third one on June 12th, which is the second Friday at 9 a.m. Pacific, 11 Central. This is where we discuss a general, again, article. It's public, but if there's any specific articles that you'd like to recommend that we discuss as a group, please, you're more than welcome to forward that along to any one of us, and we're more than happy to bring to the forefront and mention your name. Prizes may be included if you recommend a journal and we bring it up to the club. Also, this is also obviously a monthly webinar that we're attempting to do. Typically, we have it on the last Wednesday or Friday of the month, depending on scheduling. But our next monthly webinar will be on June 24th. Again, 9 a.m. Pacific, 11 Central. And again, if any topics, please feel free. We do sincerely hope that you're staying safe out there. And we do want to see you guys again. We appreciate all your questions. And of course, allowing us to be a part of your research and development. In that case, have a great weekend, long weekend if you're in the US, if you're in Canada. Stay safe, everyone. And we'll see you soon. Take care. And thank you so much.